Hey everyone, good to see you. I hear that the Wi-Fi on campus is at best spotty right now, possibly completely out. So if you get kicked out of the meeting, I'm sorry. I will make sure that I get the video, the recording of today up on YouTube as fast as humanly possible. Just so you know, Zoom takes a little while to process the video. YouTube takes a while to process the video. I usually do a little editing, cutting stuff out, but I probably won't do that for today. Just so that anybody who misses class today can see the lecture as fast as possible. So I don't know how many of us are going to make it. I'll wait another minute or so. While we're waiting, I pulled one of the checkpoint questions on the refractory period for us to use as our warm-up. Today, we're hopefully going to get into synapses. So we're going to talk about propagation of the action potential down the axon, and then talk about what's going on as our nerves signal to each other. Okay. So if we remember what a refractory period is, hopefully this checkpoint question is relatively easy for us. So when we're thinking about the refractory period, we have two types, right? We have the absolute refractory period and the relative refractory period. And this is really asking about the main difference between those two, which is whether or not we could generate an action potential. So hopefully you wrote down something like this. So when we're thinking about the absolute refractory period, we're not going to be able to fire off an action potential because this is basically happening as that action potential wave is still progressing. Our sodium channels can't be reopened during this time. Our inactivation gates are closed. So the reason we call it an absolute refractory period is this is a hard stop, a hard limit on how many action potentials in a row we can send, how fast we can fire those off. For the relative refractory period, yes, we could send off another action potential, but it's going to be hard. And that's mainly because of the potassium permeability. So those voltage-gated potassium channels are still open. We're kind of hyperpolarized. So this is happening largely at the end of that graph. As we send off the action potential, we have this period of hyperpolarization. That after hyperpolarization, because the potassium channels are open, but our sodium channels are closed. So we could fire off another action potential, but we're going to need a lot more of an impulse. We're going to need many more and bigger graded potentials in the cell in order to reach our threshold to fire an action potential. Those are more emails about the Wi-Fi. Okay. So moving forward, what we want to talk about now is the propagation of these action potentials. So when we're talking about the propagation of the action potential, I do realize that sometimes I use the word action potential to describe that whole kind of, sorry, I wish I were in the center, that whole kind of graph we have with the wave of depolarization, the peak, and the repolarization and the after hyperpolarization. But very strictly, when we talk about what the action potential is, we're talking about that spike, that depolarization to positive 30 millivolts. So I just want to clarify that a little because I know I I kind of go back and forth with how I use that word. So we're talking about how we propagate, which just means make more, basically. How we make that action potential continue down the axon. So when we're thinking about this, we want to remember that our action potentials start at the axon hillock. And I'm going to be drawing neurons a couple of times today, but now's an okay time for it as well. We'll do our first little neuron drawing of the day. So remember, when we're thinking about our neurons, we have our dendrites around the soma, which is the cell body, which is where you would find the nucleus. They funnel into this sort of pyramid, kind of triangle structure. That's your axon hillock before we send off into the our long, skinny axon. And eventually we'll have our axon terminals. Okay, so we're going to be thinking about all of these parts today. So I'm just going to put a letter. So these were the dendrites. Oh gosh, I really need this text feature. Okay, so those are the dendrites. This is the soma. This is our axon hillock. And this is our axon. And here are axon terminals. So our action potential is starting here at the axon hillock. So we had graded potentials coming in to the dendrites and the soma, and basically they all get summed together at the axon hillock. That's where we kind of care about what they add up to. If they hit threshold, we have that action potential, we spike up to positive 30. Then we're going to send them down this axon all the way out to the axon terminals. And here's where we'll be releasing neurotransmitters. Okay. So axon hillock, down the axon to the axon terminals. Okay. So basically the way this works is we have our action potential beginning in one spot. So our action potential is beginning in one spot. We're going to start at the axon hillock, and then we need to basically trigger voltage-gated sodium channels all the way down the axon. So the whole membrane of the neuron is not activated at once. When we were talking about how we get to threshold, that opens voltage-gated sodium channels, they all flow in. That was happening in a specific place in our neuron. So that was kind of a local change, not a cell-wide change. So we'll have that action potential happen at one spot right here. So we're becoming very positive in this one spot. So I'm just going to write over that sodium, just going to illustrate where we're getting to positive 30 here. And once we get really positive in this one section, those charges and that sodium starts to diffuse. So in diffusion, what I mean is they're really just spreading out. Those ions are spreading out. And so as they spread out, they trigger this next section of the membrane. So they're going to trigger voltage-gated sodium channels in this next part of the membrane to open. And so then this will also become positive, and we're going to have that wave down the axon. Does So that's what's going on, is we're triggering like this part of the membrane, then we're triggering this part of the membrane, then we're triggering this part of the membrane, all in a row, not at once. So this is going to be basically a wave of changes, a wave of charges going down our membrane. The other thing to note is part of why we care about the refractory period is that as we propagate the action potential, it goes in this direction. And the reason it can't go backwards is because of that refractory period. So once part of the membrane has produced an action potential, so this chunk, this is steps, right? So this chunk that's blue in image 1 creates an action potential, and then it goes through that whole change in voltage. So it spiked to positive 30, but then it gets hyperpolarized because of the potassium channels. And so during that hyperpolarization, we have basically this is it's, sorry guys, it's already been a long day. Okay, so as we're repolarizing, the sodium channels are inactivated, and then we have this period of hyperpolarization. So that absolute and relative refractory period both kind of act so that as these sodium charges are spreading out, they don't kind of go backwards and set off another action potential earlier in the axon. So we're going to look at this happening in patches by tracking sites and tracking charges. So we're going down the whole length of this axon. We're going to start out by looking at what happens in an unmyelinated axon. So an unmyelinated axon doesn't have the substance myelin wrapping around it. And we'll take a look afterwards at what myelin does and how this changes the propagation of the action potential. So our unmyelinated axon, we're going to have relatively slow propagation. So the conduction rate, the rate that this action potential is spreading down the axon, is slow because it has to trigger every single piece of membrane down the axon. Okay, so we're going to look at this in steps. That's the full chart, but we're going to start here. So we're starting in the axon hillock. So the beginning here of our axon, the axon hillock, the part that's connecting it to the cell body. What's happening here is we have extracellular fluid that's relatively positive, intracellular fluid that's relatively negative. We know at rest, we're at negative 70. So that's what this is showing us right now. Once we start an action potential, what we're doing is depolarizing that section of the axon. So what happens during depolarization is we have a lot of sodium flow in. So our membrane potential in that area becomes positive. So that's what we see happening here at site A. We have our region of depolarization. And you can see that at this point, site B is still negative. So site B is still at rest. It hasn't been triggered yet. It hasn't set off an action potential. Okay. What happens next is that those charges from site A kind of spread out. We see all these arrows showing that those charges are spreading out. So some of these positive charges flowed into this area for site B, got it to threshold, and so triggered off the action potential here at site B. This means that site B now depolarizes. Sodium flows into the cell at site B, so we can see that site B is then depolarized. Site A is in its refractory state, so it doesn't re-trigger, even though it's going to get sodium spreading out from site B, because during that refractory state, especially during the absolute refractory period, We can't set off another action potential. During the relative refractory period, we'd need a suprathreshold stimulus to set off another action potential, and the action potential normally flowing in its normal direction from site A to site B isn't big enough. Okay, so we see it's moved from site A to site B. Then those charges spread out. And so our action potential then triggers site C. So those positive charges at site B spread out a little bit, got site E to that threshold of -55, which means the voltage gated channels for sodium in site C opened, sodium flowed into site C. So that's what we can see happening as we go down here, propagating the action potential in a direction towards the axon terminals, but not re-triggering these areas behind because they're in a refractory state. Okay, questions about this right now? Because I know these pictures look kind of confusing.
I guess I'm kind of curious as to about how big a site is. I mean, or about how many sites might there be in a typical neuron?
So we don't really we're breaking them down into sites here for purposes of explanation, but I don't think in the neuron itself, it's really segmented like this in an unmyelinated axon. So we'd slowly kind of be doing little chunks here, but really what's happening is that it's triggering the neighboring voltage gated channels, kind of based on probability rather than based on specific sectioning.
Okay, yeah, so there's no like actual like identifiable feature in an unmyelinated neuron that's going to say like, oh, this is the end of this site.
This is site B. Yeah, I don't think so.
Okay, thanks.
It will be a little different for the myelinated sections. And that's because this is a great lead-in into myelin. So what myelin does is it wraps around the nerves. So sometimes we'll see that nerves are a little thicker, they're white because they have myelin around them. But myelin does leave gaps. So if we were looking really close, like with a microscope at a myelinated neuron, we would see what kind of looks like segments. And I don't know how many there are, I'll be frank with you. Nerves are all sorts of different lengths. But we'll see myelin around little segments, and then we'll see gaps in the myelin. So you can think of the neuron like a string, and then myelin, you just kind of tied something around it in various places. So myelin provides insulation, and we call these gaps between segments of myelin nodes of Ranvier. The reason the sectioning is important here for the myelinated neuron is that we don't actually trigger every single little bit of membrane in a myelinated neuron. In myelinated neurons, those charges still spread out, but we only trigger the action potential in the gaps between the myelin, in these nodes of Ranvier. So this means that conduction in a myelinated neuron is much faster because it doesn't have to trigger this action potential in all these sort of in-between areas. We call the way that conduction happens in myelinated neurons saltatory conduction. It basically means leaping, similar to the word for jumping in Spanish. It's like how I like to remember it. So this saltatory conduction is that we're triggering action potentials only in these spaces, these nodes between segments of myelin. Okay. So here in this image, we can see that the action potential happens to be here in the middle where we have this blue section. The action potential was previously in this red section, so it flowed from the red to the blue, And it didn't have to trigger anything here in this myelin section. So this means that in a myelinated neuron, the place where we're going to see a bunch of voltage gated sodium channels is in those nodes of Ranvier. We're going to put the channels where we could actually trigger the channels. So that's where we'll see those channels located, because we have leaps of conduction from node to node. So here is a picture similar to the previous one for the unmyelinated neuron. Now we're looking at a myelinated one. So we're still going to start our action potential in the axon hillock, because it basically is where we're adding up all our graded potentials. And we can see that this axon has myelin wrapped around it, leaving a couple little gaps, our nodes of Ranvier. So if we zoom in, we can see the action potential triggering here in our first node of Ranvier, this first gap. So we can see that there's depolarization here. We know it's depolarization because we see these positive charges on the inside of the cell. Those positive charges spread out and they're free to flow. Nothing happens until they hit the next node. That next node then gets triggered, gets up to threshold, creates that wave of depolarization. So the sodium's flowing in. So the action potential then jumps to here. So it's jumping. That's that saltatory conduction. And this is faster than if we had to trigger off every little section of membrane like we do in the unmyelinated neurons. Questions at this point about the myelinated neurons?
Are these all like different action potentials, or is it like is it multiple, or is it just like one?
We should think of it as one action potential. This is like how the action potential gets from one place to another. So it's all going to be in response to one stimulus. And that's kind of why it's a little tricky to think about what we mean by action potential, because depending on what we're talking about, we use that term a little differently. But think of it as one action potential. Think of it as the action potential was here, now it's here.
Okay, thank you.
Mm-hmm. Okay. There is another way that we can speed up conduction in addition to adding myelin, and that's that we can increase the diameter of a neuron. We'll talk when we get to the circulatory system about the relationship between the size of basically a tube and the velocity of stuff flowing through that tube. It'll become important then. But for right now, just know that if the diameter of a neuron is bigger, these charges are going to flow down it faster. Okay. So if we want to send a signal really quickly, we're going to pick a big myelinated neuron. So here we can see that there are different conduction velocities for different types of nerves, because we have nerves that do different things, that sense different things, that send different types of signals. So we can see this relationship between the speed and the diameter. So a thin unmyelinated neuron travels about 1 meter per second. So that M is meters, not minutes. So 1 meter per second, whereas a thick myelinated one can travel 100 meters in 1 second. So much faster. So you can see here that sensory nerves sensing muscle position are going really fast. They could travel 120 meters in a single second. But nerves in our autonomic nervous system, we'll talk about what that is later, we can see that they're much slower. Okay. So our next topic, we're finally getting into synapses. So the reason we were firing an action potential, the reason we were sending it down the axon is to signal to another cell. So when we're thinking about synapses, we're gonna keep in mind a couple different things. We're going to be looking at different types of synapses. So we're going to be looking at different types of signaling. We're going to be looking both, when I say different, at like where these synapses are occurring, but also what type of channels are involved and what influence that's going to have on the cells. We're going to be thinking about the speed of the responses at the synapses. We're going to think about how neurotransmitters work. And we're going to try and link this in our head a little bit to the idea of neural integration, which we talked about way back when we were talking about homeostasis. Okay. So pretty basically, a synapse refers to the connection between a neuron and a cell that it's signaling to. We're going to talk about different parts of the nervous system later. But basically we have a central nervous system and a peripheral nervous system. The central part is like your brain and spinal cord. In the central nervous system, we'll see that we have a nerve and it's going to synapse with another nerve. So we're signaling from one nerve to another. In the peripheral nervous system, we're going to see that we have nerves going out and then they're signaling to something else. So they're going to synapse with a muscle or maybe a gland. When they're connecting to a muscle, we'll call it the neuromuscular junction. So basically that's going to be a synapse between a nerve and a muscle that it's trying to trigger. Okay. For right now, we're going to be focusing on the connection between two nerves. So two nerves together. So we're going to be thinking like these central nervous system synapses. When we are talking about neurons signaling other neurons, we want to specify which neuron we're talking about, because different things are going to be happening in the neuron that's sending the signal versus the neuron that's receiving the signal. So we label them our presynaptic and postsynaptic neurons. So if we have our neuron, this is my axon, these are my axon terminals. Okay, so this is my first nerve. We might have a synapse here. So I'm going to pick this bottom one, pick another color for the second neuron. So our second neuron would then be like here, and it would go off somewhere. The synapse that we're referring to is this area in the middle. And so we would name these nerves presynaptic neuron. So before the synapse. And then we'd have our postsynaptic neuron. Postsynaptic. Post, after, synapse, postsynaptic. So this will be important so that we can clarify which neuron we're talking about. A presynaptic neuron can signal to different parts of our postsynaptic neuron. So when it's sending a signal to another nerve, it's not always going to be in the exact same place. Can connect up to the dendrites, to the cell body, or directly to the axon. So I'm going to do another drawing over here. So I'm going to be a little more specific. Right now I'm going to draw a postsynaptic neuron. So here I'm drawing some dendrites, some cell body, axon hillock, and here's my axon. So that if that's the postsynaptic neuron, we could have our presynaptic neuron coming into here. Oop, I did want to pick a different color. Okay. So we could have our presynaptic neuron coming in, forming a synapse with the dendrite. What we would call an axodendritic synapse, because it's the axon of the presynaptic neuron to the dendrite of the postsynaptic neuron. We could have an axosomatic synapse if instead we had a presynaptic neuron forming a synapse with the soma being the cell body. So if this axon of the presynaptic neuron goes to the soma of our postsynaptic neuron, that's an axosomatic connection. And kind of the trickiest one for people is an axo-axonic synapse. We will see that sometimes the axon of the presynaptic neuron goes up to the axon of our postsynaptic neuron. And we'll see that these are often going to modulate the activity of our synapses. So we'll talk a little bit more about those later. But those are our three types of synapse. They're just telling us where these two neurons are connecting to each other. The synapses can be electrical or chemical, and we're going to take a look at what we mean by that. Okay. So we're connecting neurons to neurons, or in the peripheral nervous system, neurons to effector organs, which will be muscles or glands. And we have our electrical and chemical synapses. So in an electrical synapse, what we have happening is that we have two cells, and the synapse is going to be really tight. These two cells, these two neurons are basically practically touching each other. And what we're doing is connecting the cytoplasm of one cell to the cytoplasm of the next cell with a gap junction. So a gap junction basically just opens a pore in the membrane. So anything that's in the cytoplasm of this first cell gets into the cytoplasm of the next cell. Okay, so this is basically what we're going to think about when we think about electrical synapses, because we're going to have ions traveling directly from the synapse of one cell into the synapse of the next cell. When we talk about neurotransmitters, is when we're talking about chemical synapses. And these are going to work a little differently. We're going to have a space between our two cells. So we can see here a space between our presynaptic neuron and our postsynaptic neuron. We're going to be releasing chemicals called neurotransmitters into that space, which are then going to bind to receptor proteins on our postsynaptic neuron to then have an effect in that postsynaptic neuron. We're going to look at the steps of this in detail because everything labeled in this picture is important. But first, the electrical synapses. So we have our two neurons. They're really close together. They're so close together that we're able to form a gap junction. I can't do this with my hands, even though I'd like to. Okay, so they're linked together with the gap junction, which means that their cytoplasm is basically continuous from one cell to the other. Some of these will happen between neurons and glial cells as well. Glial cells are basically supportive cells in the nervous system, and we'll talk about them later. Electrical signals between neurons are really fast, and that's because the ions can just directly flow from one cell into the next one. So we don't have any intermediate steps because those cytoplasms in our presynaptic and postsynaptic cell, they're connected. So that's really fast. These are usually going to be bidirectional communication, which is to say that, like, because our cytoplasm in one cell is directly connected to the cytoplasm of the next cell, we could have ions flow from cell 1 to cell 2, but we could also have them flow in the opposite direction from cell 2 to cell 1. I'm saying ions. We could also be talking about the flow of second messengers, which are kind of like signaling molecules inside cells, but it's easier to think about if we just focus on the ions. This means that we're not going to have just excitation or just inhibition happening in an electrical synapse, just anything at all that's in the cytoplasm of one cell can get into the cytoplasm of the next cell. So we can have both excitation and inhibition happening here. So places we see electrical synapses are the retina, the cortex of the brainstem, and the hypothalamus. So we're going to have really, really fast signaling in these areas, basically. So chemical synapses are what we're going to spend a bit more time on because they're going to be a little more complicated. So we're thinking about presynaptic neurons and postsynaptic neurons. And we talked about the fact that we can have the presynaptic neurons connecting to a postsynaptic neuron in different places. So we can see here that we have examples of each of those that I tried to draw out before. So here we see an axodendritic synapse. Okay, so our presynaptic neuron here at the top is connecting to the dendrite of our postsynaptic neuron here at the bottom. This second presynaptic neuron is connecting directly to the body of the cell here. So here's our axosomatic synapse. And here at the end, we can see a presynaptic neuron, still presynaptic, connecting directly to the axon here near the end of our postsynaptic neuron. So this is our axoaxonic synapse. Okay. In a chemical synapse, we have space between the presynaptic neuron and the postsynaptic neuron. So they're not directly connected. We're not going to have this automatic transfer of what's in the cytoplasm of one cell into what the cytoplasm of the next cell, which means that we're going to need to release things into that space and then have our postsynaptic neuron react to those chemicals that we're releasing. This is usually happening on dendrites, but it can happen in these other places. And this is going to be unidirectional communication because we're going to see that our presynaptic neuron is releasing a neurotransmitter, and then our postsynaptic neuron is receiving that signal. Okay. It's not going to go the other direction. Yeah, basically that's the point of neurotransmitters. There are dendrodendritic synapses, by the way. You could know they exist, but we're never going to talk about them. So we can forget them for the purposes of this class. Okay. So when we're thinking about the synapse, we want to make sure that we can label all the parts of our two neurons, and then we're going to see that there are a series of steps, things that happen in our neuron. So just making sure that we know where we are right now. Okay, so here's our presynaptic neuron. This is the cell that's sending the signal. So this is the cell that's going to have the action potential traveling down the axon first. So this is the very end. We're looking at the axon terminal. So remember, the axon terminal is here. This is our axon terminal at the very end of the axon. So this is the last part of the cell to receive that action potential. Okay. What we have in the axon terminal of the presynaptic neuron are something called synaptic vesicles. So a vesicle is basically a bag in the axon terminal or anywhere else. So a vesicle is kind of a bag, and in the axon terminal, that bag is filled with neurotransmitters. So our synaptic vesicles hold the neurotransmitters. So this presynaptic neuron made some neurotransmitters, packaged them, put them in these bags, and it's saving them. The way we're going to know to release those neurotransmitters, so to do exocytosis, send these neurotransmitters out into the world, by the world we mean the synapse, the space between the two neurons, is we're going to have voltage-gated calcium channels at the axon terminals. So we've been talking about sodium channels and potassium channels thus far. Now we're also thinking about calcium channels. So associate those calcium channels with axon terminals with synapses in your heads. Okay. So here we can see that voltage-gated calcium channel. Okay. We're going to have one more thing on the presynaptic neuron, but this is going to come in at the end of our process when we look at it. But just note, we also have something called a reuptake molecule present on the presynaptic neuron. On the postsynaptic neuron, so on the receiving cell side, after we have our synapse, we refer to this synapse, this space between the two neurons. We're talking specifically just about that space rather than this whole kind of systemic connection. We refer to that as the synaptic cleft. So that's our synaptic cleft, is the space between the two neurons. And then on the receiving side, the postsynaptic neuron, so that's this cell, we're going to have receptors to receive our neurotransmitters. So eventually our goal is to get these neurotransmitters out of our presynaptic neuron into this space, into the synaptic cleft, and then to react with these receptors on our postsynaptic neuron. Our postsynaptic neuron is also going to have enzymes on the membrane. You can see that that also has a triangle shape, which matches our neurotransmitters, but our enzymes have a different job. Our enzymes are going to break down neurotransmitters. Okay. So here are the steps of synaptic communication. It's a little complicated. And looking at this picture is complicated, but we're going to go through it. Okay. So when we're talking about communication at the synapse, we want to remember that what we had happening in the presynaptic neuron was we had this action potential propagating down the axon all the way down, gets to the axon terminal, which is where we are now. So we had this action potential flowing all the way down our nerve. So here's our action potential. So our first thing is that it actually gets here. That's the most important thing. So our action potential depolarizing, this wave of depolarization down the axon gets to the axon terminal. When that action potential reaches the axon terminal, so it's depolarizing this area, this opens up voltage-gated calcium channels. So step 2 is here. Calcium wants to flow into the cell. I'm just telling you that. That's true because of the specific electrochemical gradient for calcium. So calcium wants to flow into the cell. So when this depolarization from the action potential reaches the axon terminal and triggers a calcium channel to open, calcium then flows into the cell. So calcium flows into the cell, and the reaction in the axon terminal is that once we have a bunch of calcium in the axon terminal, this triggers the cell to take these vesicles full of neurotransmitters, these synaptic vesicles, and start to secrete them. When we say secrete, that means we're pushing something out, basically. We talked very briefly about exocytosis when we were talking about types of transport. So what we see happening here is this vesicle merges with the membrane here in the axon terminal. As it merges, it's joining the membrane, and then that breaks it open, pushing the neurotransmitter it was holding out into the synaptic cleft. So we can see here, step 3 is that calcium getting to the vesicle and telling it to join up with the membrane and release neurotransmitter. So here for step 3, our neurotransmitter is here. We got neurotransmitter released here. It's telling lots of different vesicles to do this. Okay. So if we have calcium in the cell, our vesicles join up with the plasma membrane, release neurotransmitter. Okay. Once we have neurotransmitter in the synaptic cleft, so in this area between our 2 neurons, we're going to see step 4. The neurotransmitter spreads out, it diffuses in the synaptic cleft. And as it diffuses, it's going to come into contact with our proteins that are in the membrane of our postsynaptic neuron. Okay, so here we can see our neurotransmitter traveling, it's in the cleft. And next, it's going to hit the receptor. So as it diffuses, it hits this receptor in our postsynaptic neuron. They didn't label these pre and postsynaptic, so I'm going to do that and make sure we know which ones we're talking about. So this was our presynaptic. And this bottom one is our postsynaptic. So our postsynaptic neuron has receptors, neurotransmitters joined to those receptors. Okay. So then what's going to happen is that this postsynaptic neuron is going to respond. We'll talk later about what that response is. It's going to depend on which neurotransmitter we're talking about. But here we have our postsynaptic neuron having some kind of a response, and that can be variable. So this basically a whole part of the pathway is the part of the synapse, the couple of steps that tell us how we're actually sending this signal. But we're not done after we've sent the signal to the postsynaptic neuron. We also have to do cleanup. Okay. So in this second and sort of group of steps, which are labeled 6, 7, 8, we're going to be getting rid of neurotransmitters. Because once we send the signal, you can see that we didn't absorb those neurotransmitters into the second cell. We just bound them on to receptors on the membrane. So they're still hanging out here for a little while. Okay. So our cleanup steps are 6, 7, and 8. One thing that's going to happen is that those neurotransmitters are eventually going to run into our enzymes on the postsynaptic neuron. So when our neurotransmitters run into enzymes, you can see this little purple junk here on the right side of the image. That's a broken down neurotransmitter. So one thing that can happen is that enzymes degrade the neurotransmitter, and then it's going to basically float out of the synaptic cleft. Another thing that can happen to get neurotransmitter out of the synaptic cleft is this part. So instead of breaking down the neurotransmitter, we can reduce, reuse, recycle. We can reuptake it back into our presynaptic neuron. So we can pull it back into the cell, package it into another vesicle, and then be able to reuse it next time we get an action potential. Okay. Questions about this diagram and this series of steps?
If they're like broken down, like in step 8, where does that go? Like, does it just go to nowhere? Or like,
Yeah, it kind of just flows out of the synaptic cleft. And then I suspect it's probably picked up by the bloodstream and excreted as a waste product, but it might be used to build other things. At that point, it's just, yeah, basically molecules floating around in that soup in our extracellular fluid.
That space in between the presynaptic and postsynaptic, is that called something?
Yeah, this space is the synaptic cleft.
Oh, okay.
Yeah,
Thank you.
so that's this word. Okay. One kind of cool thing about this process, when we think about like medications they use for mental health reasons, one class of medications, the SSRIs, which are really common for depression, for example, SSRIs stand for, what do they stand for? Right, selective serotonin reuptake inhibitors. So they actually target one of these steps. So anybody want to guess which step that they're targeting? Sorry, I know we're over time. Okay.
Step
Hmm?
7.
Yeah, exactly. So SSRIs basically stop this reuptake from working, which means that we have neurotransmitter hanging out in the synaptic cleft for longer. So it's like we have more serotonin because it spends longer in the synaptic cleft and has more time to trigger receptors, affects more receptors in total. And basically, yeah. Yep, that's what I have to say about that. Okay, you guys are free to go now.